You can see the promise that these kinds of technologies might have for us moving forward and tackling some of these big problems we are facing as humanity.
This is the Discovery Files podcast from the U.S. National Science Foundation. I'm Nate Potker. According to a U.S. Environmental Protection Agency report, only 9% of plastic waste ends up being recycled. As plastic materials degrade, which may take hundreds of years and possibly thousands in some cases, small microplastic fragments end up in the environment, in animals, and even in people, raising concerns about potential health impacts. As a result, more and more work is being done to develop more renewable polymers and resulting bioplastics that can replace traditional petroleum-based plastics. We're joined by Arpita Bose, Associate Professor of Biology, Eric Connors, a graduate student, and Tahina Ranaivoarisoa, a lab manager in the Bose Lab at WashU, where a pair of studies have recently been published looking at purple bacteria and how the PHA polymers they make might be used for bioplastics. Thank you all for joining us today.
Thank you for having us.
What is the focus at Bose Lab?
The Bose Lab has been around for about 10 years now, and we have been trying to find solutions to big problems that humanity is facing. And we are trying to use microorganisms. Not only do we go to nature to find unique microorganisms, but we also use tools of genetic engineering, synthetic biology to tame these microorganisms and ask them to do things better for us, trying to find solutions for some big problems we are facing.
So one of those solutions is bioplastics. So can you tell me what are bioplastics for the general audience that might not know, and why would the use of bioplastics be beneficial?
Yeah, so bioplastics. Why bioplastics? I think that one of the big things that we are facing right now is that petroleum-derived plastics don't degrade really rapidly in the environment. We have become extremely dependent on these kinds of plastics, and they're in everyday objects. If you look around, you find so many things that are made of plastics. And one thing we know for a fact is that the ocean ecosystems that we hear about are really suffering from these plastics, and other organisms are really affected. But in recent times, we've also heard of microplastics and how microplastics can be very deleterious for all kinds of life. So that, brings us to maybe we want to use stuff that is easily degraded, and bioplastics are a big answer to that question. And bioplastics, of course, have been explored for a very long time, but often these kinds of plastics that are on the market depend on food sources that then compete with things that, we are dealing with, like food security. So then, you really want to go to a system where you can use waste materials or things that are deleterious for us in the environment and then convert those into bioplastics and take advantage of some of these, good properties they have, like their ability to be biodegraded. So one of the things we are doing in our lab is to really look at how can we use extremely versatile microorganisms and use them to take waste materials, whether it's carbon dioxide, whether it's wastewater, and then convert them into bioplastics.
So how did you become interested in these microorganisms, in this case, purple bacteria?
Purple bacteria are beautiful. So, plain and simple, photogenic. Microorganisms are really easy to work on. They're easy on the eyes, So that was really the motivation. But truly, I think they are some of the most versatile microorganisms on the face of our planet. So thinking about solutions for big problems, there are no better microbes to work on. They're pretty beautiful, photogenic, and they could solve big problems.
So, Eric, switching to you, can you summarize your paper a little bit and tell us how purple bacteria make PHAs?
PHAs, I think, are a really good reminder that we share some pretty fundamental biology with pretty much all life on Earth, including purple bacteria. So whenever you or I eat food that's rich with carbohydrates, some of those carbs get stored away in our muscles and liver as glycogen. And then when we're really low on energy, like if we haven't eaten for several hours, our bodies will tap into those glycogen reserves as a source of energy and kind of keep things running smoothly. PHAs are basically the same thing for bacteria. And as Arpita was alluding to, we're really interested in materials that show promise as alternatives to petroleum-based plastics. And fortunately, these PHAs have some properties that make them ideal candidates. So we're really interested in learning more about how different organisms are synthesizing PHA. So moving on to my paper, we set out to characterize PHA production by two purple bacteria in the genus Rhodomicrobium. And I would argue that Rhodomicrobium are criminally understudied despite being super interesting. They have really interesting morphology. They're pretty unique among purple bacteria in terms of how they grow and divide, but they do share some important physiology that we're interested in that relates to PHA synthesis. So we thought it was a good opportunity to expand the toolkit of purple bacteria that we have to work with, and then maybe learn a little bit more about purple bacterial metabolism. So we focused on two species of Rhodomicrobium, Rhodomicrobium vanillii and Rhodomicrobium udeprense. We first combed through their genomes to make sure that they actually had the genes that you need to make PHA. And once we confirmed that, we grew them across a really wide range of growth conditions, varying the key ingredients for PHA synthesis, and then just asked how much PHA are they making. And it turns out not only do they make PHA across all of these growth conditions, but they're quite good at it compared to some other organisms that we tend to work with, particularly purple bacteria. And our data suggests that part of the reason they're quite good at accumulating PHA is related to electron conversion. Basically, it seems like they're really good at taking electrons and funneling those electrons into PHA synthesis. And this and some other literature suggests that this electron conversion is a really significant bottleneck in bioproduction, probably generally, but certainly in terms of PHA synthesis. So that tells us that as we kind of think about how to engineer strategies for enhancing PHA production by purple bacteria, we really need to think about these bottlenecks like electron conversion. And I guess lastly, I'll say it's a really good reminder that it's important to look at microbes we don't tend to think about. This is a bug that doesn't really show up in the literature. Turns out it's pretty good at what we ask it to do. So,
can you talk a little bit about that PHA cycle?
So I'll kind of zoom out a lot and then kind of focus back in. It's all about carbon. So carbon is an essential building block of life. It's the key ingredient to making biomass, and it gets funneled into lots of important metabolic processes, whether you're a person or a microbe. So whenever microbes are consuming carbon compounds, they are breaking them down into these component parts. And one of these component parts is a molecule called acetyl-CoA. And then acetyl-CoA is the main input into the PHA cycle. Once acetyl-CoA enters the PHA cycle, it undergoes a series of transformations by a variety of enzymes, and this ultimately results in the formation of these PHA granules that accumulate within the cell. If you actually look at them under a microscope, it looks like you have a cell and then these kind of big blobs throughout the cell, and those are the PHA. Later, whenever the cell is starved for energy and it needs to kind of call upon these energy reserves, it uses an enzyme called PHA depolymerase, and that breaks down the granules and releases all of that stored energy. On top of all that, there are some regulatory genes that work to either slow down or speed up the cycle. And then there are these proteins called phasins, which work to determine the size, shape, number, distribution, kind of the characteristics of the PHA granules. So that's kind of a big picture what the PHA cycle is. And all of this is really important to know because it helps us think about what are the important points within the PHA cycle that we can tinker with using genetic tools to enhance PHA production, maybe change the kinds of PHA that's being made. And that really speaks to what Tahina was able to do in his paper, where he was kind of tinkering with different parts of the PHA cycle to enhance PHA production.
So Tahina, thinking about your study, can you summarize a little bit how you were able to make purple bacteria more productive?
The initial paper we're using just the wild type Rhodopseudomonas palustris. And the main reason why we're really attracted to this bacterium is because of its ability to grow in multiple different conditions. And we were trying to increase the productivity of PHA using the ability of the bacteria to grow in a different conditions. So we tried few different genetic engineering. So we touched the gene of the Rhodopseudomonas palustris, and we tried to remove some of the gene that is naturally depolymerizing PHA. And we were thinking about accumulating more PHA by doing so. We also removed some of the competitive genes, like glycogen, which is a carbon storage pathway. Then we also increased the ability of Rhodopseudomonas palustris by wanting to fix more CO2 because it's a photosynthetic bacterium. So by increasing the ability of fixing CO2, we were hoping to increase the available carbon source for the bacterium. Then it would be naturally directed to the accumulation of PHA. So among all the genetic engineering that we made, we noticed that the increase in the ability of the bacterium to fix more CO2 was the most productive. And that's why we get really significant increase of PHA accumulation or translated into production in Rhodopseudomonas palustris.
Were there challenges genetically engineering that bacteria?
Of course, there's always a challenge, but that's also one of the reasons why we chose the bacterium, because there's a lot of tools that are already available for that kind of bacterium. Yeah, it takes some time to engineer one mutant, but it's not really like a very, very big challenge. It just takes time.
Are there challenges overall extracting PHAs from bacteria?
Yes, because first of all, the PHA is inside the cell, so you have to separate the cell debris from the polymer. We do usually organic extraction. And so if we're talking about industrial scale, that can be a challenge because of the price of the solvent and the extraction steps.
So Professor Bose, how has NSF support made this overall work possible?
Gosh, it's like done a lot. NSF stepped in, and I think that it was at a critical juncture where without their support, I don't think that Eric and Tahina would have been able to do any of this work. And they supported the rhodomicrobium work. They supported the genetic engineering and synthetic biology work. They see the promise in this, and so their support means the world. And I think that you can see the promise that these kinds of technologies might have for us moving forward and tackling some of these big problems we are facing as humanity. And I think that, NSF has a huge contribution in all of these avenues we are exploring.
So thinking about next steps and moving forward to some of those challenges, what would need to happen to make this scalable to be commercially viable?
The awesome thing is that we don't work in a vacuum, right? So I was very fortunate to have been sought after by European collaborators around the same time that some of the original publications on our bioplastic work were coming out. We were also doing basic science research to inform some of these explorations. And what I found was that many of our European colleagues are actually using wastewater, brewery wastewater in particular, and they're using open ponds and different kinds of ways of growing purple bacteria. They're in mixed populations, and they were harvesting purple bacteria biomass for bioplastics, actually. So they were doing this commercialization in a way that we were exploring in the lab setting. And I think that there's a huge synergy. Many of these places are in Spain and other countries in Europe. And I think that the hope is that, we can bring some of this thinking to the US. We are doing a lot of this groundwork, right? Of genetic engineering and synthetic biology of a natural purple bacteria. And now it's time to kind of get that to the commercialization platform. We can take, some guidance from our European colleagues, and, I meet them, I talk to them, I visit them. And so I'm going to bring some of this to the US for sure.
Right. It really speaks to how much the global community interacts with each other and can really create a nice synergy of development.
Yeah, a Spanish colleague visited my lab for the exchange, and I visited my Spanish colleague. They were doing some of this work for different things like, wastewater treatment. But there's synergies where you can see how wastewater treatment could be easily tied to bioplastic production. So that's where we are talking to each other, and there's a lot of mutual respect, and we will find solutions, and we will bring them to the global scene together.
So, thinking about how the bacteria is feeding on carbon, would it be possible to use purple bacteria for man-made carbon kind of capture applications?
Yes, that was my broad thinking when I started thinking about all of this. I was several steps ahead, and it's hard to convince funders that this is a worthwhile thing when you haven't produced a gram of bioplastic, right? But that was the hope that we can actually commercialize this stuff because it has the potential to sequester a lot of carbon. It's a carbon negative process. If you were not degrading this bioplastic, it's essentially carbon negative. If you were to degrade this bioplastic, if you're making it using the ways we are envisioning, it's carbon neutral. So I think that the hope is that we can sequester tons of carbon in different forms. And because these microorganisms can take carbon dioxide and many different kinds of organic carbon sources, that, the ideas are limitless and synthetic biology only sort of enhances that limitlessness of it. You can go kind of like, mad scientist on this and think of different ways. But the goal is to sequester carbon.
So to close things out, I'm going to go around the table here and ask each of you kind of what the next steps in your research is. So Eric, maybe starting with you, can you tell me what's coming ahead for you?
A big theme in our lab is to use basic science to inform some of these translational approaches. So we're always trying to start from first principles and understand how these microbes are doing what they're doing. What are some of the underlying genetic processes, and then how can we use that to inform these applications. So we're still in the early days of learning about rotomicrobium, for example. We're very much early days, and some things we're trying to get up and running include like genetic systems. The scale up is a big part of it for us as well. We do these things at small scales, which is great for high throughput, kind of let's tackle a bunch of different conditions, a bunch of different microbes, and try to get some quick answers. And once we have some good candidates for further exploration, then we want to scale those up and get some more biomass that we can answer additional questions with. So yeah, I think there's a lot more to be done. It's exciting.
How about you? What's coming up in the next couple of years for you?
I have been talking about those single mutant strains that we made. So we have noticed that by increasing the ability of the bacteria to fix more CO2, we have seen an increase of bioplastic accumulation. So we want to combine multiple mutations in one strain and then see if it gives more than just like one engineered strain. As Professor Bose mentioned earlier, we're really focusing on the carbon sequestration ability of Taiwan. So we want to work more toward that, like, okay, what is the best condition that we can get highest carbon fixation using the cheapest media to produce more PHP and PHAA. And then that's mostly we're going to focus on in the next coming years, and then that will be the next coming papers.
Fantastic. Professor Bose, I'll give you the last word. What are you looking forward to working on in the next few years?
Yeah, I think that the huge sort of push on artificial intelligence, machine learning, and such approaches has really opened my thinking around microbial explorations and finding new microorganisms as solutionaries. Also engineering microorganisms for different kinds of solutions. So I see my lab and me personally taking advantage of some of these approaches for sure, and getting to some of these modifications faster than we are doing right now. So trying new techniques that are available to us to really get there faster, because as a species and as a global community of organisms, we need to kind of get there a little bit faster.
Special thanks to Arpita Bose, Tahina Ranaivoarisoa, and Eric Connors. For the Discovery Files, I'm Nate Potker. Please subscribe wherever you get podcasts, and if you like our program, share with a friend, and consider leaving a review. Discover how the U.S. National Science Foundation is advancing research at nsf.gov.